Hi, my name is Tony Hanley, and I'm going to, today's date is January 10th, 2020. I'm going to be talking to you about the anatomy and enzymology of fat absorption. So I'm basically going to give you an overview of the steps in the process of fat absorption, and then I am going to go into some of the particular molecules that are used or that are created during this process. So fat is ingested in 5 forms. It's ingested in triglycerides, phospholipids, long cholesterol, long-chain fatty acids, and fat-soluble vitamins. However, the main source and mainly the focus of this lecture is in triglycerides. So that's the main source of fat in our diet. So although I'll mainly be talking about the small intestines, what we do need to remember is that fat breakdown actually begins to occur in the mouth and then continues in the stomach. However, this is just a very small percentage of the fat that gets broken down this way. A majority of the fat doesn't get broken down until it reaches the small intestines. So in the stomach, there forms chyme, which contains an emulsion of fat droplets. And the churning of the stomach helps disperse these fat droplets throughout the chyme. Now, these fat droplets have a really small surface area. They actually have a smaller surface area than particles that are of much smaller size. And this is because they are lipid soluble, obviously they're fat, and they're in an aqueous solution. So they basically have a form that tries to touch the least of the amount of the aqueous solution possible. And that is not a form that is actually really efficient for the enzymes to be able to access them so they can break down the fat. So when the chyme enters into the small intestine, it gets coated with bile salts. And what the bile salts do is it creates a small water-soluble fat droplet. And this water-soluble fat droplet is better able to be acted upon by the enzymes that can break it down, and that we can then use the broken down products to be absorbed. So in terms of digestion, the 2 enzymes that are the main enzymes of fat breakdown are lipase and colipase. Although lipase is the one that does most of the action, it can't work as efficiently without colipase. So, and they break down the lipids. Now, in terms of triglycerides are a molecule that is made up of a glycerol backbone and 3 fatty acids. And lipase actually removes 2 of the fatty acids and leaves you with the monoglyceride. So when enough digestion basically of triglycerides has occurred and you have enough of the byproducts, you get what's called a micelle, which is kind of a collision of all these remaining particles into a flat disk. And I'll get into more detail on the exact makeup of a micelle later in this lecture. But the micelles travel to an area of the lumen of the intestine known as the Busch border. So the Busch border is the apical surface of the intestine, and it contains microvilli. And if we remember from when we were learning about cells earlier in this semester, the microvilli increase surface area, allowing for more absorption, and are often found in places where absorption takes place. And in this instance, the cells that make up this membrane are made up of simple cuboidal and columnar epithelial tissue. So lipids move out of the micelle once they reach the Busch border, and then they diffuse across the cell membrane of the enterocytes. Now this just happens by simple diffusion. If you remember, plasma membranes are lipids can move freely across most plasma membranes because they are have more of a lipid-soluble surface. So they freely diffuse into the enterocyte. Once into the enterocyte, the monoglycerides and fatty acids move into the smooth endoplasmic reticulum. Within the smooth endoplasmic reticulum, they are actually repackaged to form triglycerides, the original form that we digested them in. And then they are further processed both in the smooth endoplasmic reticulum and in the Golgi apparatus to form chylomicrons. Now, if we remember, the Golgi apparatus is the membrane-bound organelle of the cell that that does create vesicles for transportation. And in this case, it creates a vesicle that allows the chylomicron to be excitosed from the cell, and this happens at the basolateral surface of the enterocyte. Now, unlike most nutrients that we digest that are absorbed directly into the bloodstream, fat's actually absorbed into the lymphatic vessels and is transported mostly through the absorbed fat is transported mostly through the lymphatic system. And this happens at what's called a lacteal, which is the villi of the lymph vessel. And so once the chylomicron enters into circulation in the lymph vessel, it circulates and it supplies tissues with fat from our dietary intake. Lipase further breaks down this fat once it gets to the tissues to create free fatty acids. And the free fatty acids are the form that our tissue absorbs. And then the tissue uses those the free fatty acids for whatever process it needs, sometimes energy consumption. It depends on the area of the tissue it's being absorbed into. When a majority of these triglycerides from the chylomicron have been broken down, you get what is called a chylomicron remnant. And this chylomicron remnant is what gets absorbed by the liver. And the liver has specific receptor sites for the chylomicron that are known as protoglycan and remnant receptors that allow the liver to uptake the chylomicron remnants. Now, the liver uses this for many different processes, but one of them is to make lipoproteins that we're familiar with, like LDL, VLDL, and HDL. So since chylomicrons have a very similar chemical makeup as these lipoproteins, it's very easy to repackage them into the other lipoproteins that the liver then circulates throughout the system. So, as we talked about bile, it is secreted from the hepatocytes, and the main active form that we were talking about is bile salts, but bile actually has three components. So it has bile salts, which is made up of a steroid acid that makes the fat water soluble. And it's amphipathic, which means it has a hydrophobic region that associates with the lipid portion and a hydrophilic side that interacts with water. So having this chemical makeup makes sense that it can then make the lipid water soluble. Bile also has a bile pigment. Its main pigment is bilirubin, and bilirubin is actually a breakdown product of hemoglobin, heme specifically. And then it contains cholesterol as well. Most of the cholesterol that is in bile is just excreted out of our system. And bile, for the most part, can be stored and secreted by the gallbladder. However, the gallbladder is not the only source of bile for our system. If it was, having your gallbladder removed would have much more serious complications for fat absorption and digestion than it does. The other pathway for bile to enter into the small intestine is directly through from the liver to the pancreas, and then through the common bile duct into the small intestines. So the enzymes of fat digestion that I mentioned earlier was lipase, and this enzyme hydrolyzes the ester linkage of fats to produce alcohol and fatty acids. So mentioned earlier, it takes 2 fatty acids from each triglyceride. It's primarily found in the pancreas, but it's also present in small intestines, in the stomach, and also within the mouth. It's secreted by the pancreas into the small intestine, and its reaction is accelerated by colipase and bile salts. So colipase is actually a protein cofactor for lipase. It is also secreted by the pancreas, and what it does is, since the lipids are being coated by the bile salts, the colipase actually displaces the bile salts, giving the lipase better access to the lipid underneath. And then we have phospholipase. It's an enzyme that digests phospholipids. Phospholipids are actually the second largest form of fat that we digest in our body. And the phospholipids get broken down and absorbed through the mucosal cells. And then a majority of them get repackaged into the chylomicron, which you'll see here in just a moment. And so they have the same path as the triglycerides in terms of being turned into a chylomicron, but they are used for other cell processes as well. And then free cholesterol is not digested, but it is absorbed, and it's absorbed on an energy-dependent membrane transport known as NPCI-LI. The micelle, so I took this picture straight from the textbook because I really liked how it showed the chemical makeup of the micelle. So it's formed of monoglycerides, diglycerides, phospholipids, and cholesterol. And as you can see from the image, you have the phospholipids have that hydrophobic tail with the hydrophilic head, and then the different molecules are scattered throughout the the micelle, and inside the micelle is those free fatty acids and cholesterol that are going to be transported to the enterocytes and then absorbed. So we have our chylomicron, which is, so it's the least dense of our lipoproteins. As I mentioned earlier, some other forms of lipoproteins are LDL, VLDL, and HDL. Each of these have a different combination of triglycerides, phospholipids, cholesterol, and protein, with HDL actually having the most protein and the least amount of triglycerides. Chylomicrons are the opposite. It has the most amount of triglycerides. It has 85 to 95% triglycerides, 6 to 12% phospholipids, 1 to 3% cholesterol, and 1 to 2% protein. And each of the lipoproteins has a protein that is specific to it, and in this case the chylomicron specific protein is APO48. And I do like this picture of the chylomicron because it once again shows you that although the lipid molecule is not water soluble, it has been coated with these phospholipids that allow it to transport through aqueous solutions like the interstitial fluid or our blood. So that's the purpose of sort of this repackaging of the lipid, is to allow it to be able to be transported in areas of an aqueous solution that it otherwise would not be able to be dissolved in. So, and here are my references. I want to thank you for taking the time to listen to me, and I hope you have a great day.